Hello guys, welcome to our YouTube channel Learning Biophysics with Team 320. My name is Abhishek Sharma and my first topic is the goal of protein physics. So the protein physics is a field of the seeks to understand the physical principles that govern the structure, dynamics and function of proteins. The primary goals of field include understanding protein structure and stability and protein stability and determining protein structures. Researchers aim to elucidate the three-dimensional structures of proteins using techniques such as X-ray crystallography and NMR spectroscopy and cryo-electron microscopy, and investigating protein stability, like understanding the factor that contribute to protein stability include the high role of hydrogen bonds and the hydrophobic interactions. Electrostatic interaction is a key goal. Exploring protein folding is studying the process by which proteins fold into their native structures is a fundamental aspect of protein physics. And the second one is exploring protein dynamics. In the exploring protein dynamics, there are characterizations, protein motions. Researchers aim to understand the various types of motions that protein exhibit, include large-scale conformational changes, local fluctuations, and vibrational motions. Investigating the relationship between dynamics and function, understanding how protein dynamics influence their biological functions such as enzyme catalyst or ligand bonding is a major focus. And third one is understanding protein function. It's first, like relating structure of function. Protein physics seeks to establish a clear relationship between the structure of protein and the biological function. The second one is investigating protein interactions. This is how proteins interact with each other to form complex and carry out cellular processes is a key area of study. Exploring protein ligand interactions, studying the interactions between protein and small molecules such as drugs or substrates, and is important for understanding protein function and drug design. The fourth one is developing applications. In first, designing new proteins. Protein physics can be used to design novel proteins with desired properties such as improved stability, catalytic activity, or specificity, and improving protein therapeutics. Understanding the physical principles that govern protein stability and function can aid in the development of more stable, effective protein-based therapeutics. Addressing protein-related disease. Protein physics can contribute to our understanding of protein-related disease and the development of potential treatments. In summary, the goals of protein physics are to unravel fundamental physics principles that underlie protein structure, dynamic, and function, and to apply this knowledge to address important biological and medical questions. Okay, guys, my second topic is conformations of polypeptide chain. So, the polypeptide chain, the primary structures of proteins, is composed of amino acid linked together by peptide bonds, and the arrangement of these amino acids in a uh, space determines protein's overall conformation, which is crucial for its functions. First, primary structure. It's a linear sequence. That the primary structure is simply the linear sequence of amino acid in the polypeptide chain. Determine the genetic code, which is determine the genetic code. And the second one is peptide bond. The amino acids are linked together by peptide bonds, which are formed between the carboxyl group of one amino acid to the amino acid of group of the next. And the second one is secondary structures. Secondary structure, in the secondary structure there are local arrangements. Secondary structure refers to local 3D arrangement of the polypeptide chain stabilized by hydrogen bonds, which is stabilized by hydrogen bonds. Common modifies two. There are two common secondary structures. Are first is alpha helix and the second one is beta sheet structure. In the alpha helix, it's a right-handed helix structure stabilized by hydrogen bonds between the carbonyl oxygen of one amino acid and amide hydrogen of another amino acid for residues down the chain. The second beta chain is a sheet-like structure formed by multiple polypeptide chains or segments of single chain arranged side by side, stabilized by hydrogen bond between the carbonyl oxygen of one amino acid to amide hydrogen of another amino acid in adjacent strand. The third one is tertiary structure. In the tertiary structure is overall folding. There are tertiary structure refers to overall 3D shape of a single polypeptide chain determined by interactions between various amino acid chains. These interactions include a disulfide bond, which is a covalent bond between cysteine residues. Second one is hydrophobic interactions. These are interactions between non-polar amino acid side chain. The third one is ionic interactions. These interactions are between charged amino acid side chains. And fourth one is hydrogen bond between the amino acid side chains, polar amino acid side chains. The fourth one is quaternary structure. There are multiple subunits. Quaternary structure refers to arrangement of multiple polypeptide chain in a protein complex. Interactions are the subunits are held together. Same types of interaction that establish tertiary structure. Factors affecting conformation. First is amino acid sequence. The first is amino acid sequence. The primary structure determining the potential conformation that a protein can adopt. And second one is environment. The environment including pH, temperature, presence of other molecules and can influence the protein's conformation. Third one is translational modifications that can occur after protein synthesis, such as phosphorylation or glycosylation, can affect conformation. And importance of conformations are like function. A protein's conformation is essential for its function, and the changes in function can alter a protein's activity or its ability to interact with other molecules. And the stability, second one is stability. The conformation of a protein determines its stability, resistance to denaturation. And the third one is disease. Misfold protein can be associated with various diseases such as Alzheimer's disease and Parkinson's disease. Okay guys, that's all. Thank you.